Hello everyone. Welcome to the first lecture of the course of the medical genetics. So today we have the introduction about the course medical genetics. I want to say nice to meet you, but so sad that is in front of my face is the just a laptop. But I will say so. Nice to meet you. So hopefully you find some very good and useful information in this group for you now and also for future. And I hope I can contribute for your understanding. So yeah, well, so I feel bad for the pandemic right now. Uh, so it's we had a medical genetic course. So at the second slide, I want to first, I want to clarify a little bit about the term of the medical genetics. So what is the medical genetics? First, I forgot. So I need to introduce a little bit about the structure of the lecture. It's quite short today. I will, like here, I will introduce the term. And the second, I will talk a little bit of syllabus and the course outline. There's some chapter every week we will cross. And then next, we will talk about the lab because of that important and also people you enjoy in the lab, wet lab, and also a dry lab to design and diagnose some disease. And I will present some several facts of the medical genetics in the world and also in Vietnam too. It's quite interesting and very hot topic right now. And I will revise some genetics milestone. I mean, it's related with history and go a little bit about the type of genetic disease and some treatment, I think, but it's very short. We'll go more detail in some chapter after this. So, well, about the term of medical genetics, at the center, so we have medical and genetic. Okay, so I like genetic more. So I talk about genetic. So who is so most famous about genetics? So we talk about the Richard Mendel, okay? Mendel, not Mandan, okay? Mendel. So Mendel is the most famous, and he's not got Nobel Prize yet, but I don't know who is older, Mendel or Nobel. But Mendel is recognized as the father of genetics because he is using the simple, actually simple now, but in his time it is very complicated, I think. So he stated three laws of genetics. So we had three laws of genetics. Mendel law, the Mendelian genetic, the law of genetic. We have the law of the dominant and recessive, right? And recessive. And another law is segregation. Sorry, about dominant and recessive. That means the one we have two alleles in the one individual in the one body. We have the two alleles. So one allele will cover the express effect of another alleles. So this is dominant and recessive. A second is we have the segregation law of the segregation. Sorry for my time on my drawing. Segregation is taught that the one baby will have the one allele from mom and one allele from dad, right? And another law is we have independently assorted. So what's that? That means two genes is located different chromosome will be independent, go into different gamete and make different offspring. But that sometimes is not true for that because of how about the two genes next together. I mean, the physically they are located in the same chromosome and they next each other. So they will go together to the next generation by genetic linkage. So genetic linkage we talk about Thomas Hull Morgan, right? So who talk about say about genetic linkage. So genetic linkage. So when I was student, I kind of, and also when I was high school student, I don't feel interesting about this at all. But actually, when I go deeper about genetic linkage, especially is very important, especially in the identify the marker for diagnose the mutation or the disease, because you cannot identify exactly what is the mutation directly make the phenotype change. You have to you only identify the marker next by there. So genetic linkage is very important concept. So talk about the genetic. We talk more about a lot of the people. We talk about the variant, genetic variant, the variants, very important. So now we go cross from the small scale to the large scale. So we have the single nucleotide polymorphism, or we call SNP. And more a little bit, SNP is quite simple, right? Because when you compare between two people, or even between two alleles in the same people, and the same location in the gene, we have a different uh, the DNA code, maybe G to C. Yeah. And SNP have the one very important characteristic for science, for medical genetics. That is they have the uh, biallelic biallelic characteristics. I will explain later. And the next is a larger a little bit. Is we have the small indel. Small indels in is an insert and there is deletion. When you compare, when you make alignment in the bioinformatic lab, so you see that we have some gap in the alignment, the picture, and some is we have some insert. That means that kind of sequence they have one or two or three or at least or five nucleotide more than another sequence, or some sequence have less several nucleotide compared with another sequence. That means we have the insert and deletion. We don't know which is the first, which comes first, and which is later. We just say that in there, and that is small because of this not big, simple. But it's larger than SNP, just only single nucleotide change. So they also have effects. So another bigger is we have a CNV. What is CNV? CNV is a copy number variation. Copy number variation. So that means is, for example, in my DNA, in that gene, I have two genes of that, and you have only one gene. Somebody has three genes of that. Or some kind of disease. Some kind of disease, for example, in Huntington disease. So in the coding region of DNA, we have the three kind of triple codon. One amino acid will be double, but many times, more than 35 times will be disease form. So this is also CMV, but small scale. So another is we have some kind of fragmentation in the chromosome scale. I mean the crossover of the chromosome in the gamete etablishment will make this gene from this chromosome we will lose and another we will gain the gene. So this fragmentation also is have some some quite the same with the CNV. And the bigger thing is aneuploidy. Aneuploidy. That means number of chromosomes in the cells of that people is different with normal case. Okay, we have a Turner is we have a less chromosome compared with normal women, and also Klinefelter we have more X like XXY compared with XY like normal men have it. So that is aneuploidy. So what is that variant is contribute for the disease because of the you see the SNP maybe you change the cause of the DNA, so the code of DNA trying to the RNA become the protein code is changed, so the activity of the protein will be changed, or sometimes it stop codon is get inside of the coding region, and then translation will be stopped, or transcription will be somehow is not normal, and they contribute to the function in the cells. And the same thing for the fragmentation or aneuploidy is have bigger effect. So more DNA variants, more DNA is abnormal, and the bigger effects will be happen. So next, so about the variants, we have two terms more. We have the germline mutation. What is germline mutation? Germline mutation is the mutation that we got from mom or from dad. That is from the previous generation. That is mom or dad, father or mom is gave us the germline. We have it from the fetus. And different with germline is we have a somatic variants. Somatic variant is is when you have the first cell and become the two cells and become the four cell A cells. So each time a copy is we have some mutation acquiring there because of the copy mechanism is in your body is not really perfect. We have a 3 billion base pair, and sometimes some base pair is not correct copy from the last copy. So we have variation will be acquired in your cells, and sometimes they will make some disease. So a lot of somatic mutation will be no harm at all, and some will make the cancer. Very dangerous, right? And you have to know that cancer is the #1 reason of death in the world right now, not HIV or any kind of the COVID or something. And what is the next one? The next one is a stroke. That means you have a big problem with the circulation system, and you feel down and die. Not you, sorry, another people. And this is number second. Stroke is also very dangerous, and now it's becoming younger. And is what information that's now is a scientist identify the stroke maybe have a contribution from the gene. Of course, maybe there's some kind of the gene related with your heart will be a mutate. I mean, it has some variants. It's not good variants. So that makes some threshold is maybe higher a little bit, or it's related with the fat metabolism in your blood. Also same effect. So right now in the medical genetic and genomic, the scientists can identify the risk of the people based on the diagnosis of your gene. So that is some information, new thing. Okay. So we talk about the variant is relate with another when we talk about genetics. So we talk about gene, right? Gene, of course. Gene. Gene is the unit of the genome. So they encode for the protein and make the function of your cells. Gene. And we have another one is the phenotype. Sorry, this panel. Phenotype. That's the outcome of your gene. But the thing is, the phenotype not only got from the gene, that also come from the environment, right? So the effect of gene and environment will contribute to the phenotype outcome. So for example, we have a twin with the genetic information between two people is identical, but they living in a different environment. Some is very good nutrition, they will get fat, and one people is very poor nutrition and they think. So the environment is contribute 100% here. But if the two people in the same environment, totally same, for example, actually we cannot make that experiment, but the same, but they still different. So that difference is caused by gene. So you know is have simple concept about gene and environment will impact to to the the phenotype. So okay, and another is not related with the variance. We talk about the now a very attractive term is the epigenetic. So you learn or you remember somewhere. So epigenetic epi is above genetic. So this term is say that so these two people is same information in genetic, but they still different because of the something happen in their cells and make the express the function of the cell is different. It means that some gene or some protein expression is not similar anymore. Why? So it's related with not AP something is we cannot explain, but we can explain. Right now, scientists can identify like that's because of the CH3 group, that methane group is it adding to the DNA code. They adding where? They adding the CG here. So they add to the C next to the G, not after. They add before. The C before G is very sensitive to get the CH3 add to the C. So why CG? Because they add by some enzyme, and the enzyme recognize CG. That's because and add to CH3 in there. So for example, if my lifestyle is not good, I smoke a lot, I drink a lot, or I sleep not enough. So my environment, I mean, it's not outside environment. This my inside body environment. The cells, my cells outside of the cell environment will change and affect to the genetic material inside of the cells and changes. So they also affect to the disease. Okay, now we kind of cover a lot of things in genetic already. So we go to the medical term. So medical is we mainly talk about the disease, right? Disease. So medical is system to care the health of the human, and the care of human is the most biggest concern is the disease. And also we talk about the also nutrition also medical relates, but it's not very big concern. But actually, the disease is the most concern. So what is the definition of a disease? So disease is a disorder in your body. Disorder in your body. So some disorder make your body is have the not good effect. So that is a we call it disease. Effects not good and become disease. Not effects become good, like become the Superman or the X-Man or the Spider-Man is a I don't know what kind of term like that. Mutant. And disease we have there we have genetic disease. Genetic disease. And another one is we have the infectious disease. Genetic disease and infectious disease. So sad that is we have a lot of disease right now. Yeah. And first we come to the genetic disease. So genetic disease is that disease is related with quite a germline, I think. The germline, I think. So the 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 germline. I mean, when we were born, we have that kind of mutant already. So we had a that's variant already, and then affect to the our body, and they express early or later at your life. And a very important concept is we have the so single gene disease. That means the disease caused by variant in the only one gene. And another term is we have the multiple gene. Multiple gene disease. So single gene disease is quite up there easy to identify and also quite easy to diagnose because of the small scale of the detection score. So it's quite easy. But actually, some kind of gene is not small. That's very big. So you have to do some some molecular technology to check your gene. It's quite a more fancy. But compared with multiple gene, is a different matter. So multiple gene is very hard to detect the signal. So you have to use a very high end technology. Very good is right now we have that kind of technology. I mean, it's a whole genome sequencing or whole exome sequencing. We can we can do that kind of stuff. So how about infectious disease? Infectious disease is also have some relationship with the genetic variants or not? And the answer is yes. Infectious disease also is have some relationship with uh, variant the variant in genetic code. Thinking about this kind of uh, COVID virus uh, disease pandemic right now. So think about in Vietnam we have nobody died yet. I don't know how about that. It's true or not, but that's in media we and the news we have it. And in China, we have just 0.5% dead. But looking for Iran, we have about 4 or 5% of infect die. So why that? What's a big difference? You can say that the Iran is maybe the system of the healthcare system is not good, but that's maybe not true. But thinking about the gene of even we are human, we are quite the same, but the genetic background of the Iran and Vietnamese and China is kind of quite different. And this virus come from Asia. I mean, east or northeast, not sure, the Southeast Asia. So maybe it's quite familiar, and our ancestor already have some gene to fight with this virus or this relative with this virus before. But Iran is no, don't have it. Remember the MERS? maybe a pandemic about 5 or 6 years ago, also in the Middle Earth, Asia, a lot of people also die. Maybe the 5% or 10% most. So so we will cover this one later. Because of the in our cell we have some kind of weapon we can fight with the infectious disease, and we can fight with the bacteria, we can fight with the viruses, another kind of disease by infectious factor. So that kind of weapon is a protein, and it made by gene, and that gene is this population and another population is different. So they also contribute to the outcome of the disease. So we cover, no, we cross, we section a little bit some terms related with medical genetics, medical and genetic. So I believe that anybody is find yourself the definition of the subject. So hope you can grab this a little bit. Okay. So next we will go to outline a little bit shortly about the course syllabus. So name of the course is medical genetics, and the code and the course type is not very obligate, and the credit about 3 lecture credit and 1 credit of the practice lab, and requirement of the subject is we have a genetic course. So but something very simple, you just remember some very simple fact of the genetic and human physiology, and I don't know about molecular and diagnostic. mm, you have it good, but it's better you learn medical genetic before you go for molecular diagnostic. But molecular diagnostic quite not only focus on human, right? Oh, sorry. And another is biochemistry. Actually, biochemistry is basic of the basic, so you have to know well about it, biochemistry. But is this level is okay. You just remember some fact about a protein and some disease related with biochemistry. I think all of the disease in our our body is come from the abnormal of the biochemistry activity in our cells. Yep, with the biochemistry you can explain it very well. And okay, and of course, learning a medical genetic also a very good thing. Not only apply for human, we can apply it for another thing, where animal term. Yeah, because of the genetic, human and animal are quite a little bit same, right? Because we are normal, and right now is the the animal welfare is only very important in our um, civilization. So you can apply some rule in the medical genetic in here to explain for the stuff in the animal welfare, I think. And about the description, so I just immersing the some important stuff in here. So the term very easy to like we apply the knowledge in the genetic to the human medical care. Yeah. And we're looking for the effect of the genetic alteration and effect to the disease outcome. Somebody is have the variation, and they have the very big effect, but the same gene they have a small effect, and we can say that. And another one is very important. We will learn about the inheritance pattern. So that means from this generation I have the disease gene, but how about my baby? He or she will have it or not? And so based on based on understanding inheritance pattern, you can explain. And then, for example, like if I got the Y chromosome disease gene in the Y chromosome, so definitely my son will have the disease, right? That's that's obviously. and and we'll cover a little bit short, I think, because of the uh, diagnostic is not very big stuff in this subject. But we also cover genetic disease diagnostic. And one is important. We will learn about the genetic counseling. What is genetic counseling? So genetic counseling is we will we will have advice, and also for the people who have the variants is I mean it's a genetic disease, and what they need to do, what we need to come to the what kind of the service they need to solve their problem. Or some people, they are carrier, that means they heterozygote of the disease, they don't have the disease yet, but if the man or the women, they try to make the baby, so they need to care about the baby. Maybe they have the high risk or they have a baby with that kind of disease. So and the sad thing is all of the almost the disease related with genetic is cannot cure, but we can get to know what kind of mutation, what kind of disease, exactly what kind of disease we have it, and we can make the life of that people is better, or somehow next generation of those people will be free of disease. That is that's a good thing. This kind of science is contribute to the society. And another is we can understand the cancer. We can understand better the cancer. So why some people have the risk of cancer, and some people is have very long life without any cancer. So we also cover that part. Very important. Very interesting. Okay. So objectives is we will understand association. So the term of association is very important between the genetic alteration and inherited disease and a somatic mutation disease like cancer. Yeah. And the principle and the practice of and the lab work related with the diagnosis of the genetic disease. So like how to that example of the contribution to this subject to your knowledge. Like you can go to outside and you see some people have some kind of the phenotype is not normal, like the very active, like he have the small hand, or he got the number 6 finger appear in the arm, and maybe some abnormal phenotype outside you can see that, but a lot of the phenotype inside you cannot see, like metabolism thing you cannot see that, but some is expressed outside you can see that. And based on that, you can check information, you can search it. Based on that, you can know what kind of disease people have it, right? And based on the symptom of the patient can tell you if you are the doctor, you are the counselor, genetic counselor. The patient talk to you, and you can check it, and you can suggest the test, genetic test people need to be carried out, and diagnose exact that kind of disease, and you can solve some problem. About the textbook, and I remember yesterday I upload the book already in the Blackboard, so you can download it and read. This book is quite good. You see that the title of the book is exactly in our object, medical genetics, right? So the book is right kind of good, and they have a lot of the example of disease and a lot of, and very detailed information, and also explain for you very for each kind of disease. They explain some kind of command and I mean the study that disease is very clear already. Some disease is still very not clear, so we don't have very detailed information. So this is related with the medical genetic book, but I also recommend you to look for the book and read the clinical genomic. You know, genetic is you focus on gene or several gene to talk about the disease. But like I said, that have a lot of factor, like the disease affects by a lot of factor, multiple gene, and you don't know what kind of gene affect to that kind of disease. So you have to use some technology, very new technology, relate with the bigger scale, the genomic scale. I mean, whole genome sequencing. I mean, any base pair in your gene will be sequenced and know it and check it. And this is the published is a very. This book is published very currently. I mean, maybe last 2 or 3 years. Very good book. I read it already. So they talk about the very new technology, a kind of effect, good thing, and some kind of obstacle we didn't cross yet. Still a lot of difficulty in the identify the genetic marker related with the disease. Okay, so this book is two book is good book, and the grading is normal. So we have the midterm, we have the 30%, final exam we have the 40%, and another we have the 30% for 100%. We had a presentation too, but I didn't decide what kind of presentation you have it because because of the late our schedule changed, so presentation will be cancelled. And we have, of course, we have assignment 50% or lab work 50%. We will talk about lab work another slide in today. And the learning outcome is also very basic and simple, like principle of human genetic. So actually, human genetic is like also is same with the what is you learn from genetics. So we talk more about the disease. We've talked more about the genome of the human, and but human genetic quite complicated right now. No, sorry. It's very complex. I think about it. 50% of human genome is repeat sequence, and a lot of part in our genome we didn't finish in the sequencing yet. Some kind of isolate in our genome is still missing. And of course we have this one general concept of the genetic and molecular mechanism of some common disorder, I mean disease. And understand the relationship between the genetic mutation and disease phenotype. Yeah, not always we have mutation and we have disease. A lot of mutation we don't have any disease. And we have the impact of the technology, recent technology on genetic, like almost related with the sequencing, and clinical medicine. Actually, the molecular technology related with the cells also very important, and they confirm the link between genetic and the disease. For example, I checked the gene of the two population. One population is disease population, and another population is healthy, and identify based on the biostatistics. So identify the mutation related with disease with a very high significance. But that evidence is not enough to guarantee my finding is correct. We still don't know that mutation relate with the outcome like the disease. We have to confirm using some model, using some molecular technology like gene knockdown. You remove that gene or increase the expression of that gene to see in the cells. And next, you have to confirm in an animal model, and then you apply that in another sample in human. That is, we can do it. And next term is based on the understanding, understanding the molecular understanding of the biochemistry of the cells. You can suggest something to treat the disease and understand the impact of the genetic disorder and their management on the patient. Uh, you will see the in fact that people have the disease, they don't want to show the disease. Yeah, for example, I had a disease and nobody know I had a disease yet. So but I don't want to show my report of disease out of other people know it, because of I know it that is a related with gene. I cannot cure it, so I don't want to share that information. You will see it later. So that is sometimes you have to have advice in a secret. And also we know some way to laboratory method to diagnose of the genetic disease. Of course, we do it in the lab. Skill is recognize the form of the some common genetic disorder, some Turner Klinefelter. In the I also still remember the figure of the boy or the girl at the Turner Klinefelter, and you know more about disease. So be able to work with patient in the genetic condition. Not only about the information about the scientific, uh, not only the scientific advice, but also the human communication advice. You can warmly talk and like the genetic counselor, the helper to people with the disorder. And having ability to work in team, that is a blah blah. You know is another subject. Same thing. Reading, present the ICIFs. The syllabus is a 15 week, and this week in number 3 already, and we are still in the first lecture. Quite late, but yeah. And I recommend you read the book to to um, to understand. It's more important to understand. And the second is you also have the ability to take care of the test. So I outline a little bit about the course. So first, like today, we will introduction about the introduce about the medical genetic. And next, number 2 and number 3 kind of simple, maybe you know already. So, but I'll also review and remind you some part about the structure and functional of gene and the chromosome related with the cells. And number 3 is related with genetic variation. This is number 3 is quite very important because of the, like I said, that they are genetic variation. We have a lot of type of genetic variation, from a small scale, from the big scale, from the middle scale like CNV or small indel. What is the impact, and also more important is the location of the variants. So they locate in the promoter, they locate in the gene body, they locate in the end of the maybe the UTR untranslated region, while they locate into the very hard to identify the fact of that, but they locate into the enhancer or the repressor of your gene, or sometimes they locate far from the target gene, you cannot quite you very hard to identify the chain. Okay, number three, finish. And the number four, quite important, is that this is we know about inheritance pattern. So the number four, we talk about the autosome, the dominant or recessive alleles. And if the disease make by the alleles, the dominant alleles or recessive alleles, they locate in the autosomal chromosome. I mean, it's from the number chromosome 1 to chromosome 22, except the sex chromosome. The sex chromosome is number 5, sex-linked disease, and non-traditional mode of the inheritance. What is non-traditional mode? That is remember, you not only have the nuclear DNA, you have another sort of DNA that comes from the mitochondria. Still, few gene is encoded by the plasmid inside the mitochondria, right? And they do very important work. They make the energy for your body. So mitochondria gene, if you have some kind of not good variant in the inside the mitochondria, so the energy system of energy power of your power is kind of limit. You don't have you don't have enough ATP to work. Is this related with your intellect or your nerve system or another function? And we'll talk about clinical cytogenetics too. I mean, it's a cytogenetic. We talk about the chromosome, the picture of the chromosome. Number of chromosome will be chain, will be double, or change some chromosome. Like we have a 13, 18, 21, and X chromosome is normally change and making disease. How about another chromosome if they change? So we have the people with the abnormal in the chromosome number of the pair of the 18, 13, 21, and X. But other chromosomes don't have it. Because of other chromosomes too important. If we have a problem with that other chromosome, we will die. We don't have that people at birth. And very important thing is biochemical genetic disorder and metabolism. The mutation relate with metabolism. But I believe many kind of the almost the mutation, almost the variant relate with the biochemical process. So number A is the G gene identification. So somebody in this class is learn about molecular diagnostic already. So in the molecular diagnosis, we know that we have the marker, and we just try to say that people have that kind of marker, that mutation or not, and we can go directly to the say that people have a risk of disease or not, right? But in here you will know it in only one chapter. It is so short. We will know the way to identify the marker, identify the gene, make the disease. It's a method the scientists use to identify that. It's too short to describe it, but we're briefly talking about that. And next thing we will talk about immunogenetics. That means some gene, a cluster of gene relate with the immune system. Very important. They will like I'm talking about some infectious disease. Our disease resistance relate with that kind of gene mutation. And also we talk about chapter 10 about the genetic basic of development. I mean some gene relate with development process. So development is also very interesting and also a mystery. Right now we don't know why the cells is programming from the for the first cell, and they will make the our with hand, our with the head, nerve, and eyes, nose very correctly without any kind of abnormal. And if normal is kind of very limited. And 11 is we talk about cancer genetic. So cancer genetic hot topic. Uh, like um, like some people is never have cancer. Some people is very rich of cancer, and also like some people is they have the inherited mutation, means the mutation got from father and mom, and but they don't have the even they have the harmful mutation yet already. But after that, why they easy to they don't have the disease that time, and they go grow and later on in the life they will got cancer. So we have some theory about that, and some multifactorial inheritance common disease. I mean, it's not a single gene anymore. We have the multiple gene, in fact. And um, 13, 14, and 15, we talk about the genetic testing, about the perspective of the genetic and precision medicine. So, I mean, it's the functional medicine. Uh, functional medicine is like um, when people is they have their own genome, and they like the drug that is quite related with the pharmacogenomic, like somebody take the course already. So the medicine is maybe some people is adapt well with that medicine, and some people is they no effect, and some people is have a harmful effect to the medicine. So they also because of the gene, because of gene, because of drug we eat the drug, and they also go in our body, go to our cells, and also digest, and they metabolize by some protein. They can cut that kind of drug. They can make the drug to another form or not. So also based on the protein, and protein also based on the gene. So you understand about the medicine, and using the genome sequencing technology, we can make the better treatment for any people. And we'll talk about clinical genetic and genetic counseling. I also talked before. So the lab outline. So I checked in the list of the student. I got some people is also maybe didn't got in the design the experiment yet. So I don't know. A lot of people I think got the course of the molecular diagnostics. So we have the extract DNA already. So with DNA extraction already. But in here now we can extract DNA from human. So I can take some hair from you, not blood, not blood. I don't like blood, and a lot of people don't like blood. Yeah. So we can extract DNA from the hair or from the inner chest in your side, and you can check it, and we can perform the some PCR. PCR can easy to conduct it. So I ordered primer already, so we can perform PCR for detect a mutation for thalassemia. So thalassemia is very very popular in Southeast Asia. Maybe can go to 5% of population. And I also design the lab for you to use your hair to check your blood type. So I know somebody maybe you didn't have the blood donation yet. So you can use your hair, a single hair, to test for your blood type using the molecular technology, and perform real time. I'm not sure about that, so I ordered some chemical we can do some real time piece of checking detection mutation, and yeah, very interesting. I mean, in my course, MedGenie kind of good thing to practice in the lab because of the interest attractive activity in the lab. Okay. So now we go to another part is um, several facts of the medical genetic. So you may recognize her. So this is Angelina Jolie. She kind of very power actress. Yeah, and in 2013, she made the world shock because she very famous. The story like this: she go to the clinic for diagnosing her gene, and she found that her gene she got the BRCA1. This is BR is what? Breast cancer number one because we have the number two. Number two also. So they the mutation of this gene is increase the risk of the development of breast cancer and ovarian cancer. So they go to another choice. So they remove, no sorry, she removed her breast. Yeah, with the women, remove the breast is really tough, not easy. But she did that. So because she's scared of the cancer development because of her gene has some problem. So you can see that. So based on this story, you can see that we had a mutation in the gene, and if you confirm with the doctor or geneticist, and they can tell you the risk, you have the high risk of this one, so you have to go to the move. The move is to go to the medical treatment, and this treatment is remove the breast for remove the risk of cancer. Yeah, they, I don't know if that's a good choice or not. But we have another concern about this kind of thing. So so we have the BRCA1 and BRCA2 location in the chromosome 17 and chromosome uh, 13, and number of the women have that mutation is quite high. But you see 55% and 65%, very high in there. But and normal BRCA 1 and 2 is only has 12%, quite low. I mean the error is high, but the number of people if women have the breast cancer is not high like that. It's quite not high. Not 50% of women have the breast cancer. So that gene is the function of that gene is they belong to the gene to protect your cells from harmful effect. So that gene is try to correct your genome. If they break BACA1 and 2, they will fix that kind of break. They are protector. They are protector of your genome. And if the protector they die, your genome will be not fixed, and you have a lot of mutation, and that a lot of mutation will develop for cancer sometime. So we have the breast cancer, we have the ovarian cancer with women, and for the men, we also have the prostate cancer. If we had a mutation in that gene, so the men only non-self if you have mutation in that gene. So some message for this story. So the good thing is from this story is the population we know more about the community, we know more about the effect of medical genetics. So mutation in BACA1 and 2 do not always lead to the cancer. And but why Angelina is designed to remove her breast? Because of the using another information in her family. The fact is, her mom also had a cancer and died because of the ovarian cancer, and she also had a mutation in that gene, and that gene is transferred to the Angelina Jolie. And another one is how scientists discover association. I mean the link between the gene and the disease. And the value of the fighting, I mean the value of fighting from the scientists. I mean, think about this. If you are a scientist, if you can identify the disease mutation, I mean the mutation related with the disease very exactly, maybe 90% or 100%. If the people got that kind of mutation, they will develop some specific disease, and you are the one find that information. You will become rich because the company will be patented and buy that invention, that discovery from you. They can make the kit, and that kit will come to a lot of customers in the world, and they can diagnose for that kind of thing. How about in Vietnam situation? Here you can see that some kind of the advertisement is already they also using the test and try to sell that kind of test for the community, so they can have seen from the baby, they can test the gene for the baby, um, and kind of diagnose what kind of ability of that baby have, what kind of important disease, or some kind of advertisement I check they can check for intelligence or some autism of the disease of the baby. But I don't know how the correct accuracies of the test. Like here we also have the detect for the cancer, detect for cancer in women, also, and detect for the NIPT, like using the blood and checking using the blood of the pregnancy women, and check for the health and the mutation of the baby. We don't need to take the cell from the baby when he or she is still the fetus. So 2 weeks ago, no, 1 week ago, I is a reviewer for the one paper. They use some next generation sequencing. I check and write some command for the for the one paper from Ho Chi Minh City organization. They conduct experiment to check the for using the NGS. I mean the whole genome sequencing. No, sorry, not whole genome. NGS using the I mean it's a large scale sequencing. We check the BAC1 and 2 for just several women have the ovarian cancer. That is I mean in Vietnam we can do that. We can do that. So I think you already have some idea about what is a medical genetic. So this is some term. So any application of genetics um, tool and also knowledge to medical practice. So we study inheritance of disease because of the mutation transfer from this generation to next generation, and we can map in the disease to specific location or chromosome. That's based on the we can know identify, like chapter A, we identify the gene related with the disease and analyze molecular mechanism through which gene cause disease. This also belong to the chapter A, but you have to know well about the protein and also know well about biochemistry and diagnose and treatment of the genetic disease. And also some is using gene therapy. Even cancer we can treat that, but the efficacy is not very high right now. We still improve. And very important in this course is talking about genetic counseling. And your job is some students before asked me about genetic counseling. With genetic counseling, you need to have a master in this stuff. Like when you go to clinic and a genetic counselor will ask and discover you some symptom and check it and no sorry symptom, and also checking your pedigree, your family have some kind of anybody have the disease or not, and you right like like you you have some relative like brother or sister have some kind of symptom, and you worry about that, and you go to clinic, and the doctor check that, and they suggest to test your DNA, some special gene or whole genome, and yeah, you will go out with sorry, you go with more information, more detail about your health in the future, right now and also the future. And also for some case like you get married, and also you get prepared for the pregnancy. Okay, this is something we know already, some milestone of in the genetics, and based on that you will that is relevant some discovery and also some invention about genetics. So this is Mendel, this is Watson Crick, this is Sanger, who is invented Sanger sequencing, and this is Kalimuris for PCR, their backbone technology in the biology, in the molecular technology. The inventor, this is I forgot his name. He's the director of the NIH, the National Institute of Health in America. So he and this one is they are competition. They doing for human genome project. Very that's the project is have a big effect to our course, medical genetics. So this is to cover list about type of genetic disease. So we have the single gene disorder, we have the multifactorial disorder, we have the chromosome disorder like whole chromosome loss or gain. We have the mitochondrial disorder, and we have the cancer like acquired somatic genetic disorder, maybe some part of your body, maybe your neck, your heart, your lung. We got the somatic mutation, and they develop a cancer. Of course, this somatic mutation they cannot go into the next generation. If they had a mutation in your sperm or your germ cells, they will go to next generation. And also we talk about epigenetic. So we'll talk about the interaction between their genetic background and the environment factor. So in here, so in the right side, we have some kind of disease, mainly because of the genetic factor. Maybe 100% here, and 11% we have the disease related with caused by the environment, and some is in the middle here, diabetic. So cysts of fibrosis normally in the European people, they got this one, and like you say, the environment, you have infectious disease. But this one is, I believe, they move a little bit related with genetic. But of course, if we got infectious disease, we have some kind of symptom with infectious symptom. But the ability to recover from infectious disease also caused by your gene, and also by environment like nutrition or treatment. So this is the list of some important genetic disease. In this list, you can see that we have some kind of name of disease in here, and this is prevalent. That means it's like in here we have Down syndrome. We have about 1,000 case. No, sorry, 1,000 people. We have one case of that. Quite high, right? Some disease are very high, like let's say hypercholesterolemia. This is less of cholesterol. Is 500 people. We had one people like that. And yeah, some people, some kind of disease very rare is in here, like Huntington disease. Huntington disease related with some your nerve cell is has some problem. Uh, Nerve cell has a problem. Your movement will be changed, it's abnormal, and remember also decrease. So, but the disease developed quite a very low. It's about one case in the 20,000 people. And you can see that
And here a lot of disease they focus on human is European with white color. Sorry. And why? Because of they are rich country, Europe, UK, or US. So the rich country, they have the project, they can check for the disease in disease and the relationship with the gene. So they also correct for Vietnamese people, but maybe some kind of level they different. So we have some room for our scientists can identify kind of the disease research research of disease in our population because they don't have information for that. But right now, a lot of international projects they try to collect the genome of any part of the of our planet uh, to to discover the different and also the similar. Based on that, they can answer better that kind of mutation really in fact for anybody or not, or just specific for the somebody in a specific population. In here we have the thalassemia. We have the around very high frequency in the South Asia. In Vietnam also very high. I heard that in the highland of Vietnam also very high population. And here we had a multifactorial disorder, like we have the stroke, cancer. Maybe 3 people we have 1 people have cancer. Very dangerous. And some diseases, no information yet. They just don't say they're rare. So we have one rule. One rule. This is a rare disease. Rare disease is a rare disease is a rare rare variance. That's variant just only in the in the in the single gene or some specific gene. And how about common gene? Common disease is also common variance. Common disease is a common variance. This is sample of chromosome abnormality. So this is Down syndrome. This is Turner syndrome. This is another one. Sickle cell disease, the single gene disorder. And this is a multifactorial disorder. Yeah. So in this week, I also just read some review. This review is they write a brief history of human disease genetics. That's also um, very new paper. Very new paper. They published about in the last month. Yeah, in the last month. And they cover from very beginning, so people is know very less about the gene due disease. But right now, the number of gene-related disease disorder is very increased. You can see that. Okay. So from just 30 years ago, less than 30 years ago, number of variant. I mean, this is the left side is the rare monogenic disease, and the right side is a complex disease with a common disease. So the information you can see that very short time, but information is accumulate a lot. So they know well a lot of gene now is from the nearby 0 we come to the 400 gene relate with the disease right now in this information. And um, because the complex disease is quite hard to identify, so it's less than that, but number is now is increase. Number increase. So now is the number of gene is about 70, right? 70,000 relay mutation is related with the complex disease. The complex disease like the diabetic cancer or something. So the most important factor make this kind of thing is the fighting, like they are also showing some kind of important milestone, like the human genome is first generate, I mean sequence, and some very big project like the next generation sequencing also very important. From that, number of the discovery related disease is the increase, and some international collaboration project between the very big country. They work together to find the information more deeper. In Vietnam right now, it also have that kind of project. So a lot of scientists try to identify the factor related with the gene data of the Vietnamese people. And this is information about uh, there's some genetic testing, and it's also the treatment. This is a gene relay with us some kind of disease in here, and gene relay, and this is treatment, and this is the different category of the disease.